Ladies and gentlemen, it is great to be with you. This is Dr. Stuart Titus, and I have a wonderful presentation today regarding the discovery and history of the human body's internal endogenous cannabinoid system. I hope you all will enjoy. Let's get started. So the ancient history of cannabis, actual fossil remains of sea squirts, show an endocannabinoid system being present in these very primitive creatures dating back to 600 million years BC. And this well predates the emergence of cannabis, the cannabis plant on the planet, which dates back to about 30 million years BC. So cannabis certainly has been used widely over the course of history in terms of medicine. And ancient Chinese artifacts discovered about 8,000 BC show cannabis use in terms of its oils, tinctures, and various preparations. Written records about cannabis use as a medicine date back to China, 2700 BC, and written records in Egypt are found at 2500 BC. In the European community, Dr. William O'Shaughnessy was treating Queen Victoria of England for her menstrual cramps with a cannabis-based topical poultice dating back to the year 1840. In the US, in the 1800 to 1937 era, cannabis was widely used as a medicine, a great part of the US pharmacopoeia, with pharmaceutical companies such as Upjohn showing 35 different cannabis-based medicines, and Eli Lilly Pharmaceutical listed 27 different cannabis medicines in their 1935 catalog. Now, doctors of that era did not know about the endocannabinoid system, did not know about cannabinoids, but they knew when cannabis was soaked in alcohol that many patients benefited from topical application or via oral ingestion of these medicines. Today, of course, we know that alcohol will leach out cannabinoids through an extraction process from cannabis and into the alcohol-based solution. In fact, in the US, in the era from 1919 to 1933, we actually had the prohibition of alcohol. So this practice by doctors of soaking cannabis with alcohol was a little bit dicey for the doctors, not because cannabis was illegal, but because at that time alcohol was illegal. Certainly things are a little different today. Now, we had the prohibition of cannabis back in 1937 in the US with the Marijuana Tax Act, but still research continued. And Dr. Roger Adams, a US-based scientist, continued to research cannabis even though there was this prohibition of its usage. In 1940, he discovered and identified cannabidiol, CBD, which he derived from what he called a red hemp oil. And his discovery led to a US government patent in 1942 on the isolation of cannabidiol. And this is US patent number 2304669. Now, cannabinoids and the endocannabinoid system. Let's take a look at these particular components of the endocannabinoid system. In 1960s, Dr. Raphael McCollum discovers THC and what he called cannabinoids. So in 1964, this Dr. Raphael McCollum from Israel isolated the THC molecule. Of course, this is the psychoactive compound found in marijuana that gives you the intoxicating high effect. Dr. McCollum determined its chemical structure and certainly looked at THC as well as CBD and confirmed Dr. Adams' work from the early 1940s of the chemical structure of these two very important cannabinoids. Dr. McCollum also called these unique to cannabis extracts cannabinoids, and he found many other beneficial health-enhancing components and compounds present in cannabis, and he termed cannabis a pharmaceutical treasure trove. In 1988, we found the endocannabinoid system and the CB1 receptor being identified. Certainly the popularity of cannabis was noticed back then by the research community, who wondered if there were special attributes of the human body that were influenced by cannabis or even these cannabinoids. And in 1988, Drs. Alan Hallett and William Devane find the CB1 receptor in a rat brain. And the CB1 receptor, of course, is influenced and responds to cannabinoids, particularly THC, which has an intoxicating psychoactive effect. The historic research paper of Drs. Hallett and Devane remains titled Discovery and Determination of a Cannabinoid Receptor in a Rat Brain. And this groundbreaking research fostered worldwide intrigue into the how and why cannabis was so popular and how it affects the human body. The CB1 receptor, the most prominent of all the brain's neurotransmitter receptor sites, is by far the largest G protein coupled receptor site in the endocannabinoid system. G protein coupled receptors are huge pharmaceutical targets. Nearly half of all of pharmaceutical medications target G protein coupled receptors. And as a large family of receptor sites, these G protein coupled receptors are involved in the expression of over 800 human genes, or about 4% of our overall human genomic sequence. Subsequent research also showed that CB1 receptors are present to a lesser degree in the gut, skin, and various internal organs. All animals with a spinal cord, going back to even the ancient sea squirt, will have CB1 receptors. And CB1 receptor signaling would prove to be crucial for regulating numerous physiological processes within the human body, including how the human's body responds to stress and how we experience pain. So the discovery of the CB1 receptor would also have huge implications for nearly every area of medical science. It opened floodgates of research into our innate cannabinoid biology. But why do we have receptors that are capable of responding to plant cannabinoids such as THC? Well, scientists then looked at the recent discovery of the opioid receptor at that time for additional clues. So the opioid receptor in the human brain were actually discovered and identified by a Dr. Candace Pert in 1973. Of course, these receptors are very strongly influenced by external plant-based opioids. Our human bodies actually produce our internal opioids, endorphins, as well as encephalinsins. Human opioids have a very short shelf life and are quickly degraded by enzymes. External or plant-based opioids have a more profound effect. They're psychoactive and in general are excellent painkillers. So scientists then wondered if the human body similarly produced internal cannabinoids. And in 1992, the discovery of the internal cannabinoid anandamide. Anandamide, or its scientific name is a very long one, N-arachidonyl ethanolamine, was the first endogenous cannabinoid neurotransmitter identified by scientists. In 1992, researchers at Hebrew University in Jerusalem, Israel, Dr. Raphael McCoolum, as well as Dr. William Devane and team, isolated this novel oil-based or lipid neurotransmitter that binds with the CB1 receptor and found this in pig brain tissue. They called this compound, chemical compound, anandamide, Sanskrit for the bliss chemical, or a term that was very suggestive of its mood-altering effects. High levels of anandamide are crucial for ovulation, and fluctuations of anandamide levels can affect fetal development. Every animal with a nervous system will produce anandamide. In 1993, a new receptor, CB2 receptor, has been identified. Present throughout the body's immune system, the gut, the viscera, the internal organs. Also present in the peripheral nervous system. CB2 receptor is also found in metabolic tissue, in many internal organs, as well as your endocrine glands. Initially reported in the journal publication called Nature in 1993, this discovery shed new light on how cannabinoid signaling regulates inflammation and why cannabinoid therapy could be so helpful for a number of treatments, a great number of autoimmune diseases. Aberrant or abnormal CB2 receptor signaling is implicated in many medical conditions such as metabolic syndrome, and this includes your heart disease, cardiovascular risk, your diabetes, as well as rheumatoid arthritis, as well as in conditions such as peripheral neuropathy, insulin resistance, liver disease, and many other inflammatory conditions. When THC binds to the CB2 receptor, it doesn't trigger the psychoactive high that cannabis is known for, because your CB2 receptors are not well expressed in the brain. So both endogenous as well as exogenous cannabinoids will cross the blood-brain barrier. Now, CB2 receptors are found in all immune cells, including the microglia and astrocytes, which modulate immune function in the brain. Though, of course, your CB2 receptors are most widely expressed in your gut, viscera, and internal organs. Anandamide, the endocannabinoid that binds to CB1, actually has very little affinity binding effect on the CB2 receptor, which means there must be another natural compound, an endogenous ligand, produced by the body that activates CB2 receptors. And in 1995, the discovery of the second endogenous cannabinoid, known as 2-AG. This study and research was found in canine gut tissue. This 2-arachidonyl glycerol, or 2-AG for short, was identified as an endocannabinoid by Dr. McCoolum and his team, as well as being identified by a Japanese scientist in the year 1995. Compared to anandamide, 2-AG is proven to be more potent, more prevalent, and more broadly expressed throughout the body. 2-AG levels in the human brain are approximately 170 times higher than those of anandamide, and 2-AG binds efficiently to both cannabinoid receptors, CB1 as well as CB2. 2-AG levels in the brain surge after a head injury or a stroke. Like anandamide, 2-AG is involved in modulating a wide range of mental and physiological processes. While they're similar and complementary in many respects, there are specific functional differences between these two endocannabinoids. Anandamide and 2-AG both protect cells against oxidative damage. Both compounds are adaptive in response to stress, but in different and distinct ways. Particularly 2-AG seems to activate this pituitary hypothalamus adrenal axis, and they are both created and deactivated by different metabolic enzymes. So anandamide mostly deals with brain communication, neurological signaling, and of course 2-AG more with immune response and the body's detoxification processes. In 1997, discovery of the metabolic enzymes known as FAAH, Fa, or MAGL, and we'll get into these here. Endocannabinoids are born and broken down by various biosynthetic and catabolic enzymes. Thanks to these metabolic enzymes, endocannabinoids are made when needed and then degraded after serving their purpose. And anandamide is broken down by fatty acid amide hydrolase, compound known as FAAH, while 2-AG is deactivated primarily by MAGL, monoacyl glycerol lipase. The molecular structure of Fa was characterized by Dr. Ben Cravat at the Scripps Institute here in 1996 in San Diego, and the following year an Italian scientist identified MAGL as the key degradative enzyme for 2-AG. By inhibiting Fa or MAGL, as CBD does, one can elevate internal and endocannabinoid levels and extend cannabinoid receptor signaling, and this has tremendous neuroprotective benefits. Variations in the genes that code for Fa and MAGL are associated with divergent health outcomes. Too much of either enzyme can disrupt or deplete endocannabinoid tone, resulting in what some might call weak constitution. Others call this clinical endocannabinoid deficiency. Now, let's look at the endocannabinoid system components that we've reviewed. We have your receptors, your ECS receptors, your CB1, CB2, your G protein coupled receptor sites, many other receptors, your vanilloid pain receptors, your organs, glands, etc., and your endogenous cannabinoids, your anandamide and 2-AG, your plant cannabinoids, your THC, CBD, CBG, CBDA, CBN, CBC, etc., and then the enzymes that build as well as break down your internal endogenous cannabinoids, your FA and MAGL. And the endocannabinoid system, of course, plays a pivotal role in maintaining healthy, stable environment inside the body despite fluctuating conditions, external inputs, and stressors. In the years ahead, new research would deepen our understanding of the components of the endocannabinoid system, an oil-based lipid signaling ensemble. Now, with the basics understood, we can now move into some concepts involving the endocannabinoid system. In 1988, the phrase the Entourage Effect first appeared in July 1988 Science publication in the European Journal of Pharmacology. The authors reported that 2-AG's binding affinity for both CB1 and CB2 was enhanced by the presence of other endogenous oil-based lipid compounds that were not, strictly speaking, part of the typical cannabinoid network. And of course, these compounds are what we've taught regularly for the past number of years. These are your terpenes that give you the characteristic smell of cannabis, or your flavonoids that give you the characteristic coloration, what makes strawberries red or blueberries blue, or the characteristic taste. Then, of course, your plant waxes, chlorophyll, etc. Dr. Ethan Russo mentioned that same year in the same journal publication the effects of the entourage compounds on the activities of anandamide, and particularly that of 2-AG. So certainly the entourage effect being known for a good number of years. And to illustrate this, we might look at seizure control with both Epidiolex, the pharmaceutical version of CBD, as well as botanical forms of CBD. Now, the pharmaceutical version is just a single, isolated, semi-synthetic, refined version of CBD mixed with a sesame oil. And here, this version is given very large doses, 400 to 700 milligrams of CBD being given daily to these very young epileptic children. Where with the botanical side, between 40 and 60 milligrams of CBD daily seems to help most of these children to overcome a significant amount of seizure episodes. And of course, looking at the side effects with Epidiolex, the pharmaceutical version, we'll see elevated liver enzymes of AST, ALT, ALP, as well as elevated bilirubin levels, which can be extremely toxic and potentially fatal. With botanical CBD, we see no such side effects, no elevated liver enzyme levels, and have done a very nice 1,000 patient study, present the results to our US FDA. When combined with cannabis chemical compound, create entourage or ensemble effect, so that the therapeutic impact of whole plant-based flower or essential oil forms of cannabis is far greater than the sum of its isolated components, as will happen with the pharmaceutical version. Now, other receptor sites, just besides your CB1 and CB2, have been identified as being influenced or being accepting of cannabinoids. In 1999, the TRP ion channels and pain receptors. So a team from Europe, and these researchers reported in the journal publication Nature, the ability of anandamide, which is normally a vasodilator, to relax blood vessels. This was mediated by its interaction with the TRPV1 vanilloid pain receptor, transient receptor potential vanilloid 1 pain receptor. And subsequent studies determined that 2-AG is also active at this receptor site, which is instrumental in regulating core body temperature and inflammatory pain. So these receptor sites, being outside the endocannabinoid system, are very accepting of cannabinoids, and particularly we see anecdotally many people using cannabis to help control pain as well as inflammation. Endogenous cannabinoids have a wider range of molecular targets than just CB1 and CB2. And here a great deal of physiological effect is seen with the family of this transient receptor potential vanilloid receptors, TRPV1 through TRPV8. In sum, there are about 65 different molecular targets within the human body outside your CB1 and CB2 receptors, and thus we can see a tremendous range of physiological effects by the use of these non-psychoactive cannabinoids. In 2001, we saw some great study and research regarding cell signaling and the ability for cells to communicate, and particularly groups of scientists publishing papers in 2001 showing that endocannabinoids engage in a unique form of intracellular communication known as retrograde signaling. Most neurotransmitters typically travel in one direction, from the signaling cell across the synapse to the receiving cell. But anandamide and 2-AG both transit in the opposite direction, from what's called the postsynaptic receiving cell to the presynaptic sender. And that's why cannabinoids, or the endocannabinoids, are referred to as retrograde messengers. They play a key role in managing how fast or how slow neurotransmitters fire. So endocannabinoids can disinhibit, or actually promote, neurological activity by suppressing synaptic activity involving GABA, which is a major inhibitory neurotransmitter. In essence, the retrograde mechanism functions as a dynamic, bidirectional feedback loop that fine-tunes synaptic transmission and cell communication. Overactive or excessive neurological function can also become toxic, and this is known as excitotoxicity. And by inhibiting the overexcessive physiological activity, reduction of the neurotransmitter glutamate can occur. Again, significant physiological benefits for the human body. Now, prior to 2001, retrograde signaling was understood to occur only during the embryonic development of the brain and central nervous system. But researchers have since learned that endocannabinoids regulate embryonic as well as adult neurogenesis, the creation of new neurons in the brain, as well as stem cell migration via this unique form of cell communication. We can think of it as like a guided missile. Once that guided missile goes off target a little bit, it starts to get a corrective signal and come back on course. And here, similarly, if cell communication provides insufficient or incorrect information, cells can start to mutate and become damaged. And here, the corrective signal, bringing that cell back to its normal, healthy state, can certainly help improve the lives of people, particularly with defective cell communication and signaling. Now, in 2003, U.S. government patent regarding the therapeutic use of cannabinoids was published, and this patent, number 6630507, was awarded to a research team including Drs. Adrian Hampson and Nobel Prize-winning laureate Dr. Julius Axelrod. The patent, titled Cannabinoids as Antioxidants and Neuroprotectants, describes a wide range of potential therapeutic benefit. And particularly for us in the CBD industry, non-psychoactive cannabinoids, such as cannabidiol, CBD, are particularly advantageous to use, according to this U.S. government patent, as they avoid the toxicity that is encountered with psychoactive cannabinoids at moderate doses, which basically means that humans can take 50 to 100 milligrams daily of CBD without any negative impairment. We can still function properly. We can drive automobiles, operate heavy machinery, etc. Where for many, just 1 to 2 milligrams of THC triggers a tremendous psychoactive effect that virtually renders people useless and has them on the couch for 6 to 8 hours at a time. Another concept emerging about this time, 2004, clinical endocannabinoid deficiency. And this was first postulated by Dr. Ethan Russo, a conventionally trained neurologist and cannabinoid scientist. He introduced this concept of clinical endocannabinoid deficiency in 2004 with a wonderful paper. And he hypothesized that diminished endocannabinoid function is at the root of several pathologies. Of course, Dr. Russo specifically mentioned several diseases, including migraine headache, irritable bowel syndrome, fibromyalgia, and chronic fatigue, as well as clinical depression, which often appear as a comorbid cluster of symptoms in patients who are cannabinoid deficient. And his research basically showed that these patients who were coming in with these very difficult to treat conditions were all cannabinoid deficient. Once he got many of these patients onto a more normal regimen of plant cannabinoids, all of a sudden these patients moved to much higher levels of health and wellness and overcame many of their challenges. And of course, subsequent studies would lend more credence to Dr. Russo's original thesis by linking endocannabinoid defects to various aberrant conditions, now including epilepsy, PTSD, autism, alcoholism, and other neurodegenerative ailments. Well, now we move on to some of the physiological processes within the human body and how these very beneficial cannabinoids can actually penetrate through the cell membrane into the cell, this intracellular benefit of cannabis. In 2005, researchers continued to discover therapeutic actions of cannabinoids and, of course, endocannabinoids as well as plant cannabinoids that are not mediated by either CB1 or CB2. A nice article in Life Science reported for the first time in 2005 that cannabinoid compounds enter through the cell membrane into what are called PPAR gamma receptor sites. And these receptors are situated on the surface of the cell's nucleus. So these cannabinoids will penetrate through the cell membrane and wind up at the receptor site on the cell nucleus itself, the brain, the inner working of the cell. So PPAR gamma is a family of oil-based or lipid-sensitive receptors, technically known as peroxisome proliferator activated receptors, and this regulates gene expression, lipid or fat metabolism, as well as energy storage. Anandamide and 2-AG both activate PPAR gamma as well as CBD. And CBD's action at this family of receptor sites, PPAR gamma, creates what's called neurogenesis, or the regeneration of new healthy brain cells, new healthy nerves, and neurological pathways. But how do anandamide and 2-AG get inside the cell? These are oily compounds, and how are these oil-based compounds able to navigate through the cell's membrane and into the cell's water-based or aqueous interior? And how do they find their way to the nucleus of the cell where they activate these PPAR receptors, which then attach to DNA promoter segments that initiate or prevent the transcription of specific genes? Well, in 2009, researchers at Stony Brook University in New York made huge strides in unraveling the riddle of endocannabinoid mobility. And here they identified a fatty acid binding protein, FABP as it's called, that transports anandamide through the cell's membrane and through the cell's watery internal ecosystem. These transport molecules also shuttle 2-AG and other lipid compounds through the cell membrane ion channels and into the interior of the cell. So these fatty acid binding proteins can direct endocannabinoids inside of the cell to the nucleus for PPAR activation or to other locations inside the cell where anandamide and 2-AG provide functional benefit. They're ultimately deactivated and broken down into metabolites by the degrading enzymes. Now, Stony Brook scientists subsequently discovered that these same fatty acid binding proteins can function as carrier molecules for both CBD as well as THC, which also don't mix well with water. When these cannabinoids hop on board this lipid transport vehicle, these fatty acid binding proteins, plant cannabinoids displace their endocannabinoids and delay the intracellular journey of these internal endogenous cannabinoids. Thus, you'll find THC and CBD activating internal components of the cell, where you'll find anandamide and 2-AG remaining near the surface of the cell longer than usual, which extends or expands cannabinoid receptor signaling. So we're improving many physiological processes within the cell while improving the overall level of cell communication by adding THC or CBD to one's dietary regimen. In 2012, French scientists reported the presence of CB1 receptors on the membrane of mitochondria, the energy-generating organelle that's within the cell as well. So another target within the cell for these oil-based cannabinoids. And this discovery shed new light on the role of the endocannabinoid system in regulating mitochondrial activity. Of course, this is crucial to how cells function. Key biological pathways that involve mitochondria, including energy homeostasis, neurotransmitter release, and oxidative stress, are modulated by endocannabinoids as well as plant cannabinoids. And researchers at the UK's University of Aberdeen show that CBD targets mitochondria to regulate intercellular calcium levels. The electrical properties of CBD also assist the cells with absorbing nutrients. Now, as reported in a publication called Philosophical Transactions of the Royal Society of London, cannabinoids as regulators of mitochondrial activity protect neurons on the molecular level. And neuroinflammatory processes contributing to the progression of normal brain aging and to the development of neurodegenerative diseases are suppressed by cannabinoids, according to this research. And thus, they may be able to influence very positively the aging process on a systemic level. So tremendous anti-aging research by this wonderful group based out of the UK and London. So binding directly to CB1 receptors on mitochondrial cells, THC will actually reduce or scale back mitochondrial activity and oxidative excess. CBD interacts with a different set of mitochondrial receptors, including what's called the sodium-calcium exchanger, which opens and closes an ion channel that facilitates the flow of electrically charged calcium ions within the cell. And the electrical activity of CBD is crucial to facilitate this calcium entry into the mitochondria, and this increases the efficiency of mitochondrial functioning. Now, Life Extension Institute has reported as little as a 1% improvement in mitochondrial functioning can add as much as 10 years to a person's overall lifespan. And regulating calcium levels inside the mitochondria is one of the mechanisms whereby endocannabinoids and CBD protect neurons and maintain cellular balance or homeostasis. In 2013, an Italian scientist, Dr. Vincent DiMarzo, again a leading cannabinoid scientist, introduced the concept of the endocannabinoid or endocannabinoidome, a complex hypersystem that encompasses our innate what's called lipodiome, or the regulation of fats throughout the human body, as well as our gut microbiome. So endocannabinoid signaling facilitates crosstalk or cross-communication between the gut flora as well as the brain, a process increasingly recognized as fundamental to human health. So this gut-brain axis has been implicated in many young children who have developed autism, and unfortunate developments in the human gut flora over time have led to these unfortunate communication errors, which then don't allow the brain to develop as properly as could be. So a lot of research now going on in this gut-brain axis, if you will. And the overall genomic sequence of cannabis, we should mention, is far more complex than that of us humans. In humans, we've identified between 20 and 25,000 different protein-coding genes, but cannabis expresses well over 60,000 different and distinct protein-coding genes. Now, a lot of current research has been going on, and certainly it's nice to mention that clinical endocannabinoid deficiency has then proven out over time So today's research is certainly taken up on Dr. Russo's initial concept of clinical endocannabinoid deficiency, and the Salk Institute's Dr. David Schubert, as well as research at Stanford University, shows that the early onset of Alzheimer's dementia is brought about by an internal endogenous cannabinoid deficiency. Dr. Schubert's research mentions that these cannabinoids will help reduce inflammation in the brain, as well as the buildup of toxic brain proteins, including beta amyloid, which is the hallmark or characteristic of Alzheimer's disease, and thus touching also on the buildup of a toxic brain protein called tau, which is the hallmark or characteristic of tremendous brain injury, or CTE, chronic traumatic encephalopathy, which is becoming the industrial disease of the National Football League. Also, research by Dr. Beth McCormick at the University of Massachusetts in recent years has done a really wonderful job in terms of digestive disorders with cannabinoids. Basically, Dr. McCormick's efforts show that the human gut is lined with cannabinoid receptors, and if one is cannabinoid deficient, the gut can't properly line itself to protect it from its surrounding environment. And this deficiency in the gut then allows the body's immune system to, unfortunately, start attacking itself. And it's only when the body becomes cannabinoid sufficient that the gut can start to line itself properly, and many people will then start to overcome digestive and gastrointestinal disorders such as Crohn's disease, colitis, etc. And in terms of COVID, interesting research has now come along from University of Lethbridge in Canada, showing that the enzymatic pathway that the COVID virus uses to gain access into the cell, the ACE2 enzymatic pathway, or the angiotensin converting enzyme pathway, can be blocked by as much as 70% via high concentration CBD, low concentration THC forms of hemp or cannabis. Recent research also from a Dr. Martha Rosner at the University of Chicago, Illinois, shows that CBD can increase the expression of interferons, and these are the human body's first line of defense against invading pathogens and viruses. Further, that CBD can reduce the cytokine storm that affects lung tissue in mid to late stage COVID patients. And finally, Dr. Rosner concludes that the patients who are regular CBD users are up to an order of magnitude, or up to 10 times, less likely to develop COVID symptoms than those who are not regular CBD users. Well, research into the how and why cannabis became so popular in the 1960s triggered a profound medical discovery in the 1980s, the human endocannabinoid system. And it was during the 1990s, so-called decade of the brain, that basic components of the endocannabinoid system were established. Since then, we've learned a lot more about the endocannabinoid system and its interaction with many other fat-based or lipid signaling molecules and receptor sites beyond just CB1 and CB2. We've learned that when the endocannabinoid system fails to function properly, plant cannabinoids can pick up the slack and provide health-restoring benefit. Today, we recognize that cannabis is such a versatile therapeutic substance because it acts via cannabinoid receptors, as well as influencing other pathways that exist throughout the brain and body. In fact, there are additional 65 other molecular targets just besides CB1 and CB2. With all the great network of targets within our bodies that are affected or influenced by our physiological processes, we believe that one can greatly improve their overall levels of health and wellness by adding plant-based cannabinoids to our dietary regimen. We've seen a progressive history of cannabis science, the emergence of non-psychoactive forms of cannabis, CBD in particular, over the past decade. Throughout its transport into the cell and interaction with PPAR gamma of the cell's nucleus, CBD creates neurogenesis, the regeneration of new healthy brain cells, neurological pathways. Additionally, CBD has been shown to target mitochondria within the cell and subsequent reversal of the neuroinflammatory processes reverses many of the characteristics of aging. And thanks to the effort of Kannaway and our family of brand entrepreneurs, the world has far greater access to health-providing benefits of CBD and non-psychoactive, non-intoxicating forms of cannabis. And we wish to establish these non-psychoactive cannabinoids as essential and vital nutrients, and someday establish a recommended daily allowance for their intake and usage by people of the world. Well, ladies and gentlemen, it's been great being with you today. We certainly wish you the best on your endeavors. Please let us know how we can help you to build a more successful business, and we certainly look forward to being with you in the near future again. I'm Dr. Stuart Titus. Thank you again for your time today.